so glad to have our Professor Jimmy Fu join us to deliver a lecture for all of us. So let's give a brief introduction of Professor Jimmy Fu. Professor Jimmy Fu is an associate professor in the Department of Psychology at Arizona State University. She received her PhD in Human Development and Family Studies from the University of North Carolina at Greensboro and completed a postdoc fellowship at Virginia Commonwealth University before joining Arizona State University. Professor Su's research focused on developmental psychopathology, gene, environment interplay, and substance use and mental health among children and adolescents, particularly in racially and ethnically diverse populations. Her works have been published in top-tier journals, including Journal of Youth and Adolescence, Developmental and Psychopathology, Translational Psychiatry, and European Neuropsychopharmacology. She is the principal investigator of an NIH R1 project examining gene, genetic, and environmental mechanisms underlying alcohol use and related psychopathology among diverse youth. And she has received multiple early career awards recognizing her contributions to developmental psychopathology and behavioral genetics. So let's extend our warm welcome for Professor Jimmy Fu.
Thank you so much. Do I need to use this one?
Whatever you like.
How are you all today?
Good, right?
Yes. All right. Thank you. It's my first time visiting Cal, so I'm very excited to have this opportunity to share some of my work with you all. Let's see. The topic here is unpacking gene environment interplay and substance use and mental health. And I recognize that on the flyer that's shared with you all, that it says taking a developmentally and culturally informed approach. And when I was preparing for the talk, I recognized that it's too much to cover everything. So I cut a lot, so I wouldn't be able to cover a lot of my work focusing on the developmental approach. But hopefully, these will be a good starting point for us to have some conversations. So I welcome any feedback and suggestions or questions for my work throughout the talk. Okay. Let me start by asking you all a question. How many of you yourself or know anyone in your social network who drinks alcohol, smokes cigarettes, or use any drugs? Only a few of you. Okay. How many of you yourself or know anyone in your social network who experience any challenges with mental health, such as depression, anxiety? Many more of you, right? When I ask this question, every single time when I teach my students or give a talk in the United States, almost every single one in the room would raise their hands. So kudos to you all, you're doing a great job. So all of my research is done in the United States with mostly people in the United States. So a lot of my work and the data that I'm showing today is based in the United States sample. Unfortunately, a lot of people suffer substance use disorders. So almost 80% of the US population age 18 or older suffer substance use disorder in the past year. So this is not just use, this is use disorder, meaning that their use, either drinking or smoking or using marijuana, is significantly impairing their functioning. So this is really a significant public health concern. And here I'm showing that across a variety of different substances, alcohol is the most prevalent. So a lot of my work focuses on alcohol use. And here's some number about mental illness. You can see that for, again, people in the United States, it's impacting a lot of people. Many people, almost 24%, experienced mental illness in the past year. So this is not only lifetime, this is fairly recent experiences. And here you can see some numbers across different types of mental health challenges. What I want you to know is that the risk for mental health challenges significantly higher for people who also experience substance use disorders. So you can see that risk for experiencing mental health challenges almost double in many cases for people who experience substance use disorder as well. So this is due to comorbidity that we often see between substance use disorder and mental health challenges. And a lot of this is due to their shared etiologies. I'm going to talk about some of those later. So since I'm here, I did a last-minute homework and looked up what is the status of mental health for people here in Macau. And it turns out that many people are affected as well. So what we're talking about today about mental health, substance use, is really an important issue that we need to be paying attention to. With that, a lot of my research, all of my research essentially, is trying to answer one question, which is what influences substance use and mental health challenges? Understanding what is the mechanisms, what is the risk factors, and what are the protective factors? What makes some people at higher risk for developing these challenges would help to inform some of the ways that we can help these people, okay? So we know that substance use disorders and mental health challenges are complex, right? They're impacted by so many things. We call that they're multifactorial, impacting by everything, literally, all the way from ourselves to our society, so societal level challenges. So my work tries to take these behavioral genetics approach, which really recognize the contributions from our genes and our social environments, and try to understand how they come together to shape these challenging, complex outcomes, okay? How many of you all have behavioral genetics background or know something about genetics? Okay, only a few of you. Then I'm on the right track. I thought I would give a really brief introduction about some of the behavioral genetics methodologies that I use in my work so that we can be on the same page. So how do we know that something is behaviorally, genetically influenced? We do these by a lot of methods. A lot of my work, some of my work use twins. As psychologists, we love twins, right? Twins come into different forms. So we have MZ twins or monozygotic twins, also called identical twins, who those twins were developed from the same cycle, so they share, the same level of genes, the same genetic profiles, and if they're raised in the same family, twin designs would assume that they share 100% of their home environment as well, right? There are another type of twins called dizygotic twins, or DZ twins for short, also called fraternal twins. These twins are just like similar siblings. They just happen to be developing at the same time, right? They share prenatal environments as well. However, like just typical siblings, they share about half of their genes. And if they are raised in the same home, we also assume that they share 100% of their home environment. So these two types of twins, they offer a really nice, beautiful, natural experimental design for us to understand genetic and environmental influences. In twin studies, what we do essentially is to collect data from many twins, MZ twins, DZ twins, and then we look at how similar these two types of twins are in terms of their phenotype. Right? For my work, we look at substance use behaviors, depression, anxiety, other mental health challenges. And if we see that MZ twins are more similar compared to DZ twins, then we can infer that genetic factors play a role in impacting these outcomes, right? And of course, if we see that DZ twins and MZ twins are equally or roughly equally similar in terms of their traits, we infer that, okay, maybe what makes them similar are not due to genetic factors, but due to their shared home environments, right? Of course, almost always, for the traits that we are interested in. MC twins, even identical twins, they're not always the same, right? And that's because there are also unique environmental factors that make them different, okay? So really, twin designs, it's about parsing out the phenotypic variance in a trait into three components, A, C, and E. A stands for additive genetic influences. C refers to shared environmental factors, the factors that they share in the home, for example, some of the parenting behaviors or families' socioeconomic status. And then E refers to unique environmental influences, any unique experiences that the twins have that makes them different. For example, they may be having different friends, right? They may be going to different classrooms, having different teachers, right? So when you hear that something is heritable, when you hear the word heritability, what we are referring to is really to what degree, right, genetic factors are accounting for the phenotypic variance in a certain trait. So we are all different, right, in how we're feeling today, whether we're drinking or not. Heritability is referring to how much of the difference among us can be attributed to the genetic differences among us, okay? Here, I'm showing you a chart that summarizes the heritability that's estimated across many studies for different phenotypes. So for things that I study, you can see that they lie somewhere in the middle. Some of you may find this surprising. For example, whether someone uses substances or struggle with alcohol dependence, right? The estimated heritability is fairly high. A recent meta-analysis estimated that it's about 50%. So the reason why people are different in terms of substance use disorder, about half of that is attributed to genetic differences. So it's really important for us to consider genetic factors in our understanding about the big picture, about what makes people at risk or resilient for these type of challenges. Okay? A lot of my current work use molecular genetic research approach. So instead of inferring the degree of genetic influences from twin designs, we actually look at people's DNA, right? That actually try to measure genetic profiles among people. So now think back to your biology 101, right? We know that humans, we have about 25,000 genes that's packed into 23 pairs of chromosomes, which, you know, includes about 3 billion base pairs, which are the building blocks of our DNA. The current genetic methodologies, most current ones, are at this level. We are mostly looking at, a lot of the work that I'm going to talk about is looking at the base pairs level, these single nucleotide polymorphisms, how we are different at one single locus on our genome, okay? Current molecular genetic research try to understand or identify which specific genetic locus or variant is impacting a lot of the complex traits that we are interested in. And currently the most popular method is the genome-wide association studies, or GWAS for short. So these looks pretty fancy, right? But essentially it's very simple. Essentially it's scanning across a whole human genome, right? Usually it's, these GWAS studies would involve testing more than 1 million genetic alleles and testing how it's associated with a certain phenotype that we are interested in. Could be substance use disorder, could be anxiety, could be major depression. Essentially, it's running 1 million or more regression analysis, right? Looking at how how many risk allele you have on a certain locus is associated with an outcome that you're interested in. This is a really different approach. If you're familiar, years back or a couple decades before, genetic researchers are testing one single gene or candidate gene, and they're guessing that they're thinking that that gene is having an impact on a phenotype. But GWAS studies takes a hypothesis-free approach. They're like, I don't know which gene matters because we don't have enough knowledge about that yet. So we're gonna look at everything along the genome, okay? And we're gonna do a hypothesis-free approach, testing every single allele across the genome and see which one stands out, okay? But the idea is really simple regression analysis, but run it for 1,000,000 times, okay? Of course, with that many times, you have to correct for multiple testing, right? Because if you run it for so many times, something is gonna become significant, right? So with that, when we do GWAS studies, we use really small p-values to correct for multiple testing. And here, this is a beautiful chart that we call Manhattan plots. This is not showing very much, but think about this as the towers, high, tall buildings in Manhattan, New York, right? Every dot on these Manhattan plot represents one single genetic allele, okay? And then the ones that's above this red line is showing statistical significance at the genome-wide level, having a p-value of 0.0080 in between, okay? And it's significant at the genome-wide level. And this chart is showing the genetic, the results from a GWAS study with more than 1,000,000 people. Most of those are from European ancestry, so white people in the United States, or in Europe, or in Australia. And people of other ancestry are represented as well, but at a much lower proportion. What I wanted to say in this graph is that based on a series of GWAS studies that has been done for virtually any trait that you're interested in, it's been done for so much. But the story is consistent, which is that these complex traits, psychiatric traits, behavioral traits that we are interested in are polygenic, meaning that they are not only influenced by one gene or a few genes, they are influenced by many, many genes. Each of them have a very small effect. And you can see that in this example for problematic alcohol use, genes show up across almost every chromosome, right? And then you can see that it's not only one dot that's above this line, it's many, many genes, okay? Here I'm giving you another example for major depression. This most recent, currently the largest GWAS study on major depression with also very large sample size, showing that so many genes is having an impact on these complex traits. So that's the basic genetic methodologies that we use in our work to characterize or understand that genetic actually play a role. But I'm a psychologist, I'm not a geneticist. What I'm really interested in is really what's happening next, right? So yes, of course, we all have our genes. Every one of us in this room carries some sort of genetic risk for something, right? And that's okay, because genes are really not our destiny. What's really interesting and important is to know that environmental factors really matters. And it matters in part by modifying our genetic risk. Even people having high genetic risk are not necessarily destined to develop problems. That's because their environmental experiences can play an important role in modifying those genetic effects, okay? So I'm going to show you a few examples of gene-environmental interaction effects. I always like to show this one because this study was published by Danielle Dick, who was my postdoc mentor. This is one of the early studies using a twin design to show that parenting behaviors, I know some of you are interested in parenting behaviors here, research here, showing that parenting behaviors can play a really important role in modifying genetic influences. So I want you to pay attention to these blue line here, representing additive genetic influences, which we were talking about based on twin design, right? You can see that as parental monitoring increases, estimated genetic effects decrease, right? So parental monitoring here, we are referring to the degree to which parents know about their adolescents, in this case, whereabouts, who they hang out with, what they're up to, right? So when parents are monitoring their kids or have a clear knowledge about what their kids are up to, genetic factors, genetic influences on substance use decrease. You can think of it as adolescents may not have as much opportunities to engage in substance use here, and so their genetic disposition for these type of behaviors have less opportunity to be manifested, okay? Here is some of my work. This is actually my dissertation work many years back. It's focusing on a candidate gene, which is no longer, the state of the science, but I wanted to show it because it also shows a really clear example of the fact that parenting can matter by buffering genetic risk. This study focused on 5-HTT-LPR, which is a gene that involved in serotonin. It's called serotonin transporter gene, which involve brain functioning, particularly related to how we regulate our emotions, our mood. Here, the y-axis represents people's risk of following a heavy drinking trajectory based on their data from age 12 to age 32, okay? Compared to their risk of following a really low risk or light drinking trajectory. You can see that for those who carry one or two of a risk allele for this gene, right? These people are higher at genetic risk. You can see that having higher parenting quality offers that risk. When parenting quality is very low, you can see that genetic factors, having that genetic risk is associated with much higher risk of following a persistently risky, heavy drinking trajectory among these young people. But when parenting quality is high, you don't see that risk, right? It's buffered. But of course, candidate genes is problematic. Not problematic, but the method itself is not ideal because it's only focused on one gene. And I just showed you that these genes are these phenotypes are really complex. It's not only influenced by one gene, right? And so current molecular genetic work is increasingly using what we call a genome-wide polygenic score approach, which is trying to leverage discoveries from GWAS studies and to apply it to target sample to calculate a polygenic score that represents people's genetic profile, capturing all of their information across their genome. Okay, so essentially what this works is that a GWAS study would estimate effect sizes of each particular genetic locus on a particular phenotype. And when we calculate polygenic score, we essentially are calculating a weighted sum score, right? Okay, effect size of gene 1 or allele 1 is, let's say, 0.2, and this person carries 2 of those risk alleles. So it gets 2 multiplied by 0.2, and then for the next genetic allele, you continue to add it up, and then a GWAS, a polygenic score essentially is capturing that weighted risk along the whole genome. And when you have that score in any population, it's going to have a very beautiful normal distribution, representing low risk all the way to high risk. So in this room, every one of us will sit somewhere, most likely in the middle, for polygenic risk for any trace that we're looking at. Okay? So my current work used this approach to test gene environment interplay. Here I'm showing one example from our work that look at how genome-wide polygenic score for alcohol dependence predicts alcohol use behaviors among European American and African American adults, and how their environmental factors, in this case social support, can moderate that risk. So here we show that those people who report that they perceive that they have strong support from their friends, these adults, their genetic risk is buffered. So these dotted lines in both graph here represent those people who have high friend support that they self-reported, and those lines are not significant. So polygenic risk scores are no longer predicting higher risk for their alcohol use behaviors when they experience high social support. And one more example, this is done recently by my PhD student, Bilal. He was interested in looking at community environment, so where we live, right, how we perceive our neighborhood or community also can matter. So in this example, community cohesion, which is representing, this is parents self-report on how they feel about their neighbors, their community, whether they feel safe, whether they feel that they can trust their neighbors, whether their neighbors are helpful, right? When you need something, can you knock on the door, right, next door? So here, again, this is looking at major depressive disorder polygenic scores, showing that people who have higher polygenic scores for major depression, right, in general, they are having or experiencing more depressive symptoms, which is consistent with what we would expect based on findings that genetic factors play a role in depression. However, what here Bilal is showing is that community cohesion can attenuate that risk, right? Having higher degree of community cohesion, that risk gets attenuated. So this line here, the slope is less steep, okay? So those are a few examples. Collectively, a lot of work in this field has shown that gene-environment interaction is a phenomenon that is quite, widespread and impacts almost all the traits that we are studying, including education attainment, externalizing behaviors, alcohol use, school belonging, everything that you can think about. It's a really widespread, common phenomenon in psychology research that has been demonstrated. A lot of the research has shown two patterns of gene-environment interaction effects. One is that of social control versus opportunity. The idea that in environments where there's stronger social control against a particular proper behavior to occur, usually in those kind of contexts, genetic influences will be lower. Okay? When there's more opportunity for certain proper behavior to happen, genetic influences tend to be stronger. So research shows that higher parental monitoring, right, that is exerting more stronger environmental control, social control against proper behaviors. Genetic influences are lower. On the other hand, among adolescents who hang out with a lot of peers who engage in deviant behaviors or who drinks a lot of alcohol, for example, genetic influences tend to be stronger. Okay? Another pattern is that of contextual triggering or diathesis stress. This is showing that usually under stressful experiences or circumstances, genetic risk for mental health challenges are likely to be amplified. So people who have higher degree of genetic risk, if that risk is coupled with adverse, stressful experiences in their life, they're more likely to develop mental health challenges. Okay. Now I wanted to pause a little bit and ask you all, do you think that environments are purely environmental? We're talking about genes, we're talking about environment. Do you think environments are just environments? One of the questions that we are also asking in our work is whether environmental factors themselves are also genetically influenced. We ask this question because it really matters. A lot of the interventions efforts focus on the environment and assuming that by changing that environment itself, it can work for everyone, right? But what if the environment itself is not randomly distributed? What if people's genetic profiles also play a role in what kind of environments that they experience? Then if you only focus on the environment, then maybe that intervention will be less effective, or maybe it will only be effective for some people, but not for others, depending on their genetic risk profiles, right? So, the answer to that question is yes, right? Here I'm showing a, almost 2 decades ago now, there's one systematic review by Ken Keller, who is a prominent behavior geneticist. What they show is that across all the environmental, quote-unquote environmental factors that they look at in their review, a lot of this is based on twin studies, showing that all of those environmental factors are heritable to some degree, okay? It's not too high, right? It's lower than what we just saw for alcohol use disorder, for depression, but it's there, right? It ranges on average from 15% to 30% based on their review. And the environmental measures that they include in their review include things like, parenting, family interactions that we study. It includes things like stressful life events, social support, peer interactions, a lot of things that I study, many of you may be interested in as well, right? So environments are not just environment to some degree. A few years back, we published a paper looking at adolescents' reported family interactions and look at to what degree are they heritable and whether that heritability changed over time across adolescents as well. And as you can see here, we're showing that on the left side is relational support. This represents positive aspect of family interactions. So feelings of warmth, support, and love. On the right hand is relational tension, so family interactions that involve conflict and tension. As you can see that, yes, they're heritable. Estimates of heritability are largely similar for males and female adolescents. However, what we're showing here is that it can change over time. Estimated heritability for late adolescents on family interactions are fairly high, some of it reach 0.5. So as adolescents get older, how they perceive their family interactions in part are largely driven by themselves, right? They are playing an increasingly important role on their genes are playing an important role in contributing to variations in perceived family interactions. Outside of the twin research framework, our recent work using polygenic scores also are showing patterns of what we call gene-environment correlation, which is essentially correlations between people's genetic predisposition and their environmental experiences. So our work using data from mostly the adolescent behavioral cognitive development study with US adolescents with large samples with about 11,000 people. We have shown that, for example, higher polygenic risk for externalizing problems. It's associated with higher family conflict, lower parental acceptance. So there's more conflict and parents are less loving for adolescents who have higher genetic risk for externalizing behaviors. This polygenic score is also associated with higher risk for peer deviant behaviors and lower peer prosocial behavior as well. And similarly for major depression polygenic risk, we also found that it's also associated with higher family conflict. So these are a few examples showing correlations with gene environment, between genes and environment using molecular genetic methods. We tested mediation models and looking at the effect of polygenic scores, even beyond accounting for their phenotypic behaviors of externalizing behavior, for example, we still show these effects, these correlation there. And gene-environment correlation has been documented as one potential mechanism for how genetic factors may contribute to developmental outcomes among people. So this is also referred to as environmentally mediated genetic influences. So gene-environment interplay, our correlation can occur through multiple mechanisms, right? Passive, our genes and our environment could be correlated simply because we inherit genes from our parents, and our parents can also provide some of the environments for us. So through that, it can be correlated through a passive mechanism, right? It can also be correlated through active gene-environment correlation, right? We have our genes, and genes influence some of our traits, what we like, and we actively seek out our environment. If you're social, outgoing, you seek out social context that's different. You may go party more, right? That's you playing an active role in shaping your environment. Evocative gene-environment correlation can mean that who we are can impact how people interact with us, right? A shy, quiet child may attract different responses from people compared to a really outgoing social child. So gene-environment correlations can occur through multiple mechanisms, but what's important to note is that these patterns are really demonstrating that genetic differences are really associated with systematic differences in people's environmental experiences and exposures. So they're not random, right? And this is also mean that in traditional psychology research that focuses on looking at, the link between a certain environmental factor and a certain outcome may overestimate the impact of environmental effects. Okay. With that, I want to just briefly transition to my most current work, my passion, where my passion lies, is to tackle the diversity problem in behavioral genetic work. Okay, so a lot of the work that I just showed you, particularly, or including both twin studies and the molecular genetics studies, including GWAS and polygenic score work, a lot of those significant findings are based on results with samples of European ancestry of white people in the United States, UK, Australia, etc. And there's a huge diversity problem in behavior genetic work, meaning that people of non-European ancestry are hugely underrepresented in this kind of work. So this figure here shows, on this hand, it shows the proportion of people who are included in GWAS studies. And here shows the distribution, global distribution of the population by ancestry. And the red represents European ancestry, right? You can see that they're highly overrepresented, and people of other ancestral backgrounds are underrepresented here. Why does this matter? it matters because the current methodology of applying GWAS findings to calculate polygenic score to characterize people's genetic profiles, right, can be biased and inaccurate and not working so well for other people if they were not represented in GWAS. And that's because people of different ancestral backgrounds can be different in terms of their distributions along their genome. And if we are applying findings from GWAS of people of European ancestry background to, let's say, East Asian population, African ancestral population, the ancestry in discovery sample and the target sample is not matching. And so the polygenic scores would not work as well. The farther away the distance in terms of their ancestry, the less predictability of those polygenic scores. And this is one of the major reasons why a lot of the work that we do when we look at, when we try to translate findings of polygenic scores to African American populations, Latin American populations, Asian American populations, is not working so well. So that's problematic, okay? Of course, there are some progress. People now in the field has recognized the problem, and there's ongoing efforts to try to tackle that. In the United States, there is a NIH-funded large effort called All of Us. It recruits millions of people across diverse backgrounds, collecting genetic data, environmental data, and try to tackle this problem. And the Psychiatric Genomic Consortium also have a cross-population working group try to tackle this problem, to include people of other ancestral backgrounds in this work to increase the representation. However, the problem not only lies in the genetic side, right? We know that these traits that we are interested in are influenced by both genes and environment. And only tackling the diversity problem from the side of the genes is incomplete. So the other side of the diversity problem is also very true. The fact is that a lot of the work looking at gene-environment interplay, including those that I just showed you, right? They only focus on a few environmental factors. They focus on, here is some data from a meta-analysis that published a few years back, focusing on reviewing studies that look at polygenic risk scores and interaction with environmental factors in predicting substance use outcomes. Here are the environmental factors that they focus on. They focus on intervention, they focus on adverse life events, parenting, peer relations, a few others that often are parental education, socioeconomic status. But of course, human development is impacted by many more environmental factors beyond the few that has been looked at, right? Many of you may be very familiar with this model, Bronfenbrenner and colleagues' bioecological model of human development. Really, it's highlighting the fact that human development is impacted by environmental factors across many, many levels, right? Ranging from our immediate social environment, like our family, our friends, all the way to the macro systems, the culture, social policy, etc. And current GBIE work really has only looked at a few of them, and I think that's problematic. In Bronfenbrenner's model, culture is placed at this outer circle, right, as part of the macro system. And Bronfenbrenner and colleagues argues that culture can impact human development indirectly through impacting the more immediate environments. However, other scholars has argued that we may need to reconsider that, right? Particularly for scholars who study the experiences of minoritized populations in the United States who are not white, who are immigrants, for example, like myself. argue that culture is way more important than what Dr. Bernard originally was thinking about. It's not laying out there, and they're arguing that culture is everywhere. It's an inherent part of our life. We should be positioning culture into the microsystem, or reconsider that. And they call this model the cultural microsystems model, and really is arguing or suggesting that culture is impacting everything we do. It's in our everyday practices. It impacts where we go, what we do, our values. Okay. What I want to say is that culture is important and should be considered, particularly when we try to understand the experience and developmental outcomes of minoritized populations, okay, or of diverse cultural backgrounds. There are other theoretical frameworks that specifically have been developed for understanding development of racial ethnic minoritized children in the United States. And for example, this model, the integrative model by Garcia-Cole and colleagues, shows that our social positions, right, our race, our social class, even our gender identity, can really impact developmental outcomes through complex pathways. And here I wanted to highlight just a few, right? It shows that, for example, our social positions can impact whether we would experience discrimination, right? In the United States, really unfortunately, racial ethnic minoritized youth often experience racial discrimination. They are treated unfairly by the majority population, and that is a really widespread stressor that can amplify genetic influences. Okay. Of course, this model, I really love it because it doesn't take a deficit approach to understand our development, but also recognizing that there can be adaptive cultural experiences that can protect this youth development as well. For example, cultural values and traditions. One example that's been studied a lot is that of familism. So among Asian American families and among Mexican American families, there's a strong cultural value of emphasizing the importance of family, right? Families hang out together, support each other, and that strong cultural value has been shown to be really protective for a lot of developmental outcomes. But this model also highlights that even for quote-unquote environmental factors or family processes that's often studied in G/E work, we can also take a cultural lens to understand those processes as well. A lot of those research on parenting look at typical measures like parental support, warmth, acceptance, monitoring, psychological control, right, without any adaptation or nuanced consideration about how those constructs may play out in diverse cultural populations. Or maybe there are other aspects of parenting that is more salient or relevant for some cultural groups than others. For example, a lot of immigrant or minoritized populations in the United States, families or parents would engage in what we call parental cultural socialization practices. That is an important aspect of parenting behavior that has never been explored in gene-environment interplay work. But they are important, right, for the cultural groups that needs to be represented in gene-environment interplay work. So a lot of immigrant parents, including myself, would engage in cultural socialization, meaning that we actively socialize our children about our culture, our heritage culture, where we come from. For example, in Chinese New Year, which is upcoming soon, we always celebrate our traditional holidays. We teach our children about Chinese culture, socialize them so that they can be proud of being Chinese while also living in a different country. So this is an important aspect of parenting behaviors that has been largely ignored. Justin, just one example. So to that end, our work in my research team really tries to address the diversity problem from not only the genetic side, but also the environment side. So I'm just going to share, this is one last data that I'm going to share that we just recently published last week, I think. We use twins, also from the ABCD study, that at this time we only focus on those who identify as minoritized racially. So Black, Hispanic, Asian American, etc. And we look at racial discrimination, which is, again, a prevalent experience among these adolescents, and it's a really well-documented stressor that can increase risk for substance use and mental health outcomes. This is the twin design, again, here focusing on this blue line here, representing genetic influences or estimated genetic heritability. What you can see that is for both internalizing and externalizing problems, estimated genetic risk or genetic influences for internalizing and externalizing behaviors among these minoritized youth are increasing as the self-report experience of discrimination increases. So again, discrimination as a stressful event, right? Stressful experiences amplifies genetic influences on these outcomes. Okay. With that, we, my team, and myself, really believe that taking a culturally informed approach to GE research is going to be really important. This includes increasing representation, of course, but also really transitioning to also emphasize these really salient social-cultural experiences of people with diverse racial, ethnic, cultural backgrounds, taking a really broader consideration of what we conceptualize as E is really important. Okay, this is not easy. Okay, as a psychologist, I come to this work with my background under focusing on environment, but a lot of behavior geneticists, they are trained as geneticists. They are not really nuanced in terms of their thinking about environmental influences. So moving forward, this field really is emphasizing interdisciplinary collaborations. We recently developed this framework to help people to show what we're thinking about how broadening the environment. So recognizing that not only the typical social environmental context is important, like family, peer, neighborhood, but also recognizing social processes that includes both access, positive, protective processes, but also cultural stressors like discrimination or bicultural stress, the stress coming from navigating to different cultures as well. I didn't get to talk about this circle here today, but our recent work also is showing that even structural and institutional environments matter. So one of our recent paper looked at the state level across the United States, whether state level policies are against immigrants, that state level policy factor variations across the different states in the United States is predicting suicidal outcomes among minoritized girls. So in states where they have policy that is not very friendly to immigrants, those minoritized youth would have higher risk of suicidality. So even these seemingly really broad, not immediate social environments can matter. So we are arguing that it will be important to broaden up our conceptualization of E in this work. Okay? With that, I would just want to pull all this together and let you all know why this matters, right? Why do we care? Why do we want to understand gene environment interplay, particularly if we are not going to change our gene, right? At least not just yet genetic editing is not ethical, right? But what does this all mean? Can we do anything about it? What does it mean for interventions? This paper was published a few years back by one of my best friends. So I'm biased here showing her work. But here it shows a beautiful story, which is, although while polygenic scores might increase risk for alcohol dependence in this case, an intervention that focuses on parenting in this case buffers that risk for adolescents. So adolescents who goes through an intervention with their parents that are targeted at promoting positive parent-child relationships, communications, parental monitoring, we don't see a significant impact of polygenic risk for their alcohol behaviors. So interventions matter, and understanding gene-environment interplay tells us that even for people with high genetic risk, this is a high-risk sample, there's hope, right? Interventions can still buffer that risk. And there's a systematic review focused on how the interactions between gene and intervention, and consistently shows that story as well, meaning that interventions can really buffer genetic risk. And the flip side of that story is that interventions don't work equally for everyone, okay? If you don't account for people's genetic profiles and assume that it works for everyone, the answer is that that's not true. Interventions may not work for some people, depending on their genetic profile. One caveat is that currently polygenic scores is not doing a great job. By that I mean that it's not accounting for a large proportion of the variance in the traits that we are interested in. It's predicting something, but the predictability is not great. Usually, the best polygenic scores so far is predicting somewhere between 5 to 10% of those outcomes, and some are even predicting less of variance in these outcomes. And all this is to say, and in one of our paper that we published a couple years back, shows that really at the current state, polygenic scores are not ready for clinical use to identify people with substance use disorder and mental health challenges. So it's not ready for clinical use yet. You cannot go in and say, tell me my polygenic risk so that I can know, I can identify who is going to develop depression or anxiety disorder. We're not there yet. But I think even with that, the story is going to change, because in a foreseeable future, it might be more and more relevant for all of us. So here, the data is a bit old, but I'm sure if I get recent data for today, it's going to continue to increase. This is showing the number of people who gets their DNA tested. How many of you have heard about 23andMe? So 23andMe is a genetic testing company in the United States. Literally, you can, if you purchase their product, they're going to send you a kit, usually a tube, and you can spit into it, provide some of your saliva, and then they would test your genome, and then they would give you your genetic information back. And this is showing how many people are doing that. Okay, it's becoming very, very popular. This is the price for how to do it currently. It takes $199 US to get your DNA tested, and they come back, the delivery to you, what you're buying with this money is your genetic ancestry profile, okay, and also your health profile based on your genetic information, polygenic scores included, okay. So you will get polygenic risk for yourself for a series of outcomes that you choose, okay. People are very interested in getting this information. And a recent study a few years back, not so recent now, by my postdoc mentors group showing that young adults are highly interested when they survey people. Close to 80% say that they are very interested in receiving genetic feedback about their alcohol use disorder possibility. Okay. This is, I showed this is to say that it may be part of the future, but the challenge is right there, right? People who guess their genetic risk not necessarily know how to use them and how to interpret them. So there's a lot of ethical challenges there. How to give genetic information in an ethical way that is actually helpful instead of creating harm is a big question. But there's ongoing work happening, and all that is planning to get into this field as well. It's considering, given that people are really interested in genetic feedback, and the field of genetics has really advanced so significantly that it's going to be in the foreseeable future, right? Personalized medicine is a goal in the United States and across the globe as well. With technology advances, we want to be able to leverage them carefully to actually serve people. So a lot of research is focusing on how we can give back personalized genetic information in a helpful way. And research is showing that by accompanying that genetic feedback with educational materials that can be helping people understand that genetics is not destiny, right? Understand the complex gene-environment interplay is involved in who they are, right? And giving genetic counseling and helping them to, based on their genetic profile, making healthier life choices, maybe, right? Be aware of your risk and knowing that your outcome is driven by a complex combination of your genes and your social life experiences may be helpful in serving to leading to changes in behaviors that may be able to help promote healthier outcomes, both behaviorally and emotionally for people. So with that, I'll just quickly summarize what we just talked about. Really, I wanted to convey to you that substance use outcomes, mental health outcomes are really complex, right? They are impacted by complex interplay between genes and environment. And those interplay come into different forms. One is gene-environment interaction, meaning that environmental factors can really either exacerbate or buffer that genetic risk. We also show you that genetic factors may influence developmental outcomes by influencing our exposure to specific environments. So our environments are not randomly assigned, okay? We may be systematically are exposed to different environments, in part depending on our genes. Of course, there is really important need for us to increase representation of minoritized populations in this work, but also take a really broader conceptualization of the E in this work as well. There's potential, right, for integrating genetic information in interventions. I say potential because I want to be cautious. There's literally a lot of ongoing debate on how to do this in a healthy way, do it ethically, do it in a way that would not cause harm. So, but I think this is an ongoing area that is going to be very important because genetic testing is right here. It's going to become more and more popular. It's going to become part of everyone's life in the future. Okay. With that, thank you everyone for your attention, and I welcome any feedback. This is a beautiful picture of my lab at ASU, and I acknowledge my funding mechanisms and my investigators. Thank you so much.
Thank you so much for the great and thought-provoking talk. So a few years ago, we actually seek collaborations with Professor Su in conducting some research using ABCD data set, right? But it just has so many barriers in my mind, and so I think we can get started on that collaborations later on. But it provides a lot of insight for me in terms of course, the research, but also for teaching. Well, out of coincidence, actually, I was teaching the educational psychology yesterday, and we are talking about gene environment interaction. But after hearing this talk, I realized I was talking so superficially. So there's a lot to add to the slides, right? So I will work on that.
So invite me to come back.
Yeah, of course. Yeah, definitely. So any questions? Like, we are open for questions. Yeah, sure.
Thank you, Professor Zhou, for this very interesting talking.
Professor Su.
Oh, Professor Su. Sorry. I'm a little nervous. And I'm surprised that gene can even influence the environment. So I'm wondering if when they study the environment, do they distinguish the self-report environment and the objective environment? Because I'm thinking that maybe the genes may just influence the perceptions of environment or the way we just view the world rather than the objective environment. Thank you.
Really great question. I think evidence for gene-environment correlation has been shown for environmental factors based on different measurement methods. Although you made a great point that the estimates, I would guess, it would be much higher for self-report or perceptions of the environment. Think about gene-environment correlation. You may not think, how do our genes impact that? I think my talk today is only superficial in talking about that, but another missing picture is a big box in between genes and environment, right? Genes are just genes, right? It impacts what? Our body, how it makes proteins, and then it impacts how our brain functions. So there's a lot of intermediate phenotypes there. And I think even with objectively measured environments, I'm not sure, thinking about parenting that you are interested in, let's say we welcome kids to the lab and then we observe parent-child interactions and your behavior coding their interactions. That's not self-report, right? But do you think that's not going to be impacted by parenting? My guess would be that it would, right? Because kids, their genetic profiles impacts who they are, or maybe their personality or their behavioral characteristics, and that would impact how their parents are interacting with them, right? That's one mechanism that we were talking about is the opposite gene-environment mechanism. But I think you made a great point that the estimates would likely differ depending on how you measure those environments and what specific environments that you're looking at. At the societal level that I was talking about, at the last, state level policy, of course not. I wouldn't think that our genes would have that kind of impact. But for immediate social context, our interactions with families, friends, school teachers, I would think, that would play a role, regardless of how we measure it.
Yes. Thank you.
Okay, thank you for your sharing, Professor. You mentioned that the importance of the intervention. So I'm interested, like, do you have any recommendations regarding the timing for implementing the intervention?
Earlier the better. That is a short question, short answer, but it's also true. Early intervention is always better than later intervention, and that's shown in a lot of our work as well. That's the part that I didn't talk about, taking a developmental approach. So things that we study, like substance use, right? It doesn't occur one day. No one just wake up one day and realize that they struggle with substance use disorder. It has a developmental trajectory, usually manifested early on, with some people, even starting in childhood with higher risk for externalizing behaviors, for example, then they are later on at elevated risk for substance use disorders. And so a lot of our focus is trying to understand how the mechanisms manifest earlier in life to inform early intervention. The intervention by polygenics for intervention study that I show, that's done by one of my good friends, is an early intervention as well. Not too early, but it's in early adolescence, right at the time when many adolescents start experimenting alcohol use. They don't wait until they already are struggling. That they would be called treatment or intervention. Prevention is better than intervention, and intervention is better than treatment in terms of cost effectiveness. So yes, in the field of intervention and prevention science, which by the way, ASU has one of the leading institute focusing on that, it's consistently shown that the earlier, the better. Of course, there's nuances, right? Some things it may work later in life, and it depends on the outcomes. And some phenotypes may not be early onset, and it's hard to, identify people at risk. But overall, the broader picture is that early intervention, early prevention is better.
Thank you.
Well, thank you very much, Professor Su. I think it's a very, very interesting topic, and I'm amazed by the fact that environment is kind of genetic or systematic as well. And I was wondering, well, to me it sounds a little bit desperate, like also you try so much things and still the environment or like family stuff, peer stuff is kind of genetic. And at the current stage, I was wondering, so what are the takeaways for us as educational practitioners? So, like, so much things are genetic, so can we still do something?
I get the same question when I was giving my job talk, I remember. A professor was joking and saying, oh, so what I have been doing in my last few decades, looking at parenting influences in child development don't matter? I'm like, no. Rest assured it matters, right? And particularly, it's shown by the G by D effect, right? It shows that even for people who are at high genetic risk, environments matter in buffering those risks. And what we are really saying is that we need to be cautious in thinking about environments are not randomly assigned. It's not purely environmental. So when you are trying to, focus on intervening a child's parenting or home environment, you need to not only focusing on creating interventions targeting on that environment itself, you also need to pull in the child and consider the child and the parents' genetic profiles. While genetic profiles is hard to conceptualize in an intervention setting, a lot of work is showing that even without genes, you can know that there are some genetically influenced traits like, for example, for adolescents, impulsivity, right, for substance use. There are certain degree of personal characteristics that are genetically influenced. You can consider them, right? You can imagine designing a family intervention program that would look differently for a child or a family that is high in terms of risk-taking, in terms of sensation-seeking, impulsivity levels, compared to a family that is more, socially withdrawn, let's say, right? Or not as outgoing, or experience more challenges with, different kinds of teens that you can characterize to make you desperate. Environments are genetically influenced, but the degree of that is relatively low. And that side of work is really gonna tell us that we want to be able to interpret our findings that only focus on environmental, to be, more cautious, and also be recognizing that if you design an intervention that only focuses on environments and not thinking about individual differences, that intervention may not be equally effective for everyone, right? So you need to be considering both sides, getting more to the personalized part of it.
Okay, so we are out of time. So thank you. Yeah, thank you all for coming. Yeah. Thank you.